everyone, welcome to my show. My name is Elnaz. I have a PhD in cellular and molecular biology, and I'm a bioinformatician. As you know, I discuss different subjects related to RNA sequencing data analysis. In this video, the subject is comparison of alignment tools for analysis of RNA sequencing data. Well, as you know, we have lots of tools for alignment in RNA sequencing data analysis. And for this video, I found a really good article which is called Comparison of Short Read Sequence Aligners Indicates Strengths and Weaknesses for Biologists to Consider. It's been published at 2021 and in Frontiers of Plant Sciences journal, and I thought this is a really good article and I should add the information in my presentation and also discuss the related issues. So we have lots of alignment tools. In this article, a few of them which are considered as the most popular ones have been compared, and I also mentioned them. You know that we have BOT2, BWA, HiSat2, MOMR4, STAR, and TopHat2. And we want to compare them based on a few metrics. What are our metrics? Well, many of us know, or for the people who haven't done an RNA sequencing analysis, I should mention that alignment part or step of the RNA sequencing analysis process is one of the longest processes. It can take a really long time, like 24 hours or even longer or shorter. This is based on your computer and also your alignment tool. So one of the metrics should be the runtime. Another one should be the efficiency, because as you know from our previous video, the efficiency of alignment depends on how much tools can align the short reads against the genome or transcriptome as the reference. The first matrix is alignment rate. In this diagram, the percentage of reads aligned has been drawn against tools. And as you can see, we have 4 levels of alignment tools. The first level is BOT2 local alignment and BWA, which showed the highest percentage of aligned reads. The second level belongs to MOMR4 and STAR. They showed more than 75% and less than, again, I guess, 80% of aligned reads. At the third level, we have BOT2 end-to-end alignment and HiSat2, which showed less than 75% of aligned read. And at the last level, or fourth level, we have TopHat2, which shows a really, really low percentage of aligned reads. I should mention one point in here that TopHat2 has been updated to HiSat2, and today we usually use HiSat2, not TopHat2. But the article mentions that TopHat2 is used as a reference for this comparison. So we also mentioned that like this. The second matrix is runtime per read, or let's say the race of the alignment tools. We want to see which alignment tools works faster and which works slower. Let's take a look at this diagram, in which the y-axis shows runtime per read as microsec per read, and the x-axis shows the tools. So we have 5 levels of the alignment tools. The first level, let's say the slowest alignment tool, is BOT2 local. Yeah, this is reasonable, let's say, because it's local alignment. And you, as you know, or maybe you should read a bit about local alignment. This is a really slow process. The second level is MAMR 4. The third level is BWA, and the fourth level is BOT2 end-to-end alignment tool. STAR covers for both third level and fourth level. And the last level, or let's say the fastest alignment tool, is HiSat2. So what's the rate for this? HiSat2 takes less than 10 microseconds per read for alignment. It's a really short time. Consider that we have billions of nucleotides in a reference genome, and we want to align one short read against all those genomes. And this takes less than 10 microseconds. So how long is the run for the slowest one, which is BOT local? The slowest alignment tool, or BOT2 local alignment tool, takes about 45 microseconds per read. So there's not much difference when we take a look at this and we consider the microsecond. But if you consider that in a library of short read for each experiment in which we have a few samples, it will take a lot of time. Also considering that the difference between the slowest one and the fastest one is like the HiSat2 takes 4 times slower or less time than BOT2 local. So this is a good metrics to consider when we are designing our experiment or analysis. The third metrics is transcriptome coverage. Also in this diagram, you can see in the y-axis the transcriptome coverage as a percentage and minimum alignment length in the x-axis. There's a one point in this diagram. Almost all of our alignment tools work better in the minimum alignment length less than 1,000 nucleotides. This is a fact. So for reads longer than that, like 2,000 or 2,500 nucleotides, which one works better? A short review shows us that STAR and HiSat2 works better than the other tools, and it has a bit better transcriptome coverage, but still there's not good, because the transcriptome coverage shows about 10% and the other tools show like 5%. But let's take a look at the shortest length, like shorter than 500 nucleotides. So we can see that we have more than 70% of transcriptome coverage, and they are still similar. I mean, all alignment tools show similar performance for transcriptome coverage when its minimum alignment length is shorter than 500. Also, there is a special range of alignment length between 500 and 1,000 of nucleotides. So let's take a look at this. In this comparison, we can see that BWA and BOT2 end-to-end, and also BOT2 local, and MAMR 4 show as a intermediate one 50% of transcriptome coverage. But STAR and HiSat2 show more than 500%, 50%, excuse me, it's like 60%. And when we want to compare the performance, when it comes to the reads longer than 500 nucleotides, it's an important factor to consider, because the average performance is 10% higher in star and high SAT2. But in shorter reads like 100 nucleotides, etc., or 200, BWA and BOTI2 end-to-end alignment shows better performance. So this is a really good metrics to decide about which alignment tool you want to use for your own RNA sequencing experiment, because it shows you that you can decide based on your own experiment. What's the length of your reads? How long are your short reads? Or what's the experiment design? Like, are you using a nanopore sequencing, which produces long reads or short reads? So this is up to you and your experiment, but these are good metrics to decide about them and choose the best alignment tools. There's another metrics, which is let's say a tricky one, distribution of unmapped transcripts. So what this shows to us? As you can see in here, we have let's say the functions or the genes involved in the functions which are distributed in different parts of the genome. For example, RNA processing and modification, chromatin structure dynamics, cell cycle control, etc. And we have two groups. One of them is uncategorized and another one is no results. So let's say these are the unmapped transcripts for each of the most modern alignment tools: BWA, BOTI2 end-to-end, STAR, and TopHat2. As you can see, the lowest number of unmapped transcripts belong to BWA. The second one is BOTI2 end-to-end. The third one is STAR, and TopHat2 shows the highest number, so that TopHat2 has unmapped transcripts number like 15 times more than BWA and about 8 times more than BOTI2 and like 3 times more than STAR. As we mentioned before, TopHat2 is considered as a reference or primary alignment tool, but you can see that how modern alignment tools show the progression in efficiency. Also, I will let you know that the highest range of unmapped transcripts belong to replication, recombination, and repair genes, or genes which are involved in these functions and distributed in the genome. The other one is function unknown, and also the function unknown is important. But also, I want to mention one point for you. If you have watched our previous video, you know that we have two types of alignment tools. One, alignment against reference transcriptome, and the second one is alignment against reference genome. As you know, STAR works for alignment against genome and also transcriptome. BWA works only for alignment against genome. The advantage of alignment against genome is that we have reads which belong to introns, let's say, and we have the opportunity to align them when we are doing alignment against genome. But this doesn't happen when we do alignment against transcriptome. And somehow we can see that STAR shows a bit more number of unmapped transcripts in function unknown. Maybe these are the reads which belong to introns, which are not involved in reference transcriptome. I mean, this is an assumption, this is a theory, but it could be close to the reality based on the rules or let's say the algorithms of each alignment tool, and also the advantages or disadvantages of each alignment tool. There's also another thing that has been assessed in this study: the average runtime per read vs average alignment rate for modern aligners. So in here, the y-axis shows time on 5 cores per read as microsecond per read, and x-axis shows the percentage of aligned reads. As you can see, we have the highest percentage of aligned reads for both tool local and BWA. But still, the time also for both of them is really long, especially for both T2 local. As I mentioned at the beginning of the video, this is reasonable because of it's a local alignment. But for BWD, let's say it's been adjusted. The second long runtime is MUMR2. The similar percentage of aligned reads is observed in STAR, but STAR shows the shorter runtime per read, so we can, let's say, conclude that BWA and STAR show a good, or let's say the optimized time per read against the percentage of aligned reads. But I should mention that again, that you should consider your own experiment and decide about this. What's your preference? Do you prefer to have the highest aligned reads and also a few other questions, or you prefer it to be a quick alignment? So this is up to you. Here I collected the results for all of the assessed alignment tools. As you can see, the percentages are, let's say, close somehow, or when we are considering all the metrics together, there's a more neat result about which tool I should use for my own experiment. But I also should mention this. Before deciding, making your last decision for choosing the alignment tool, it's better to answer these two questions too. One of them is that which tool can help you for finding the splice junctions. This is about which tool works, how works each tool, and what are the outputs of the tools, and let's say the advantages of some tools. And also, which tools work better for quantification if your experiment, the goal of your experiment is quantification. So you have to decide about this. Thank you for paying attention to my presentation. Please like this video if it was helpful for you and share it with other people. Also, to watch more videos which will come every Thursday, subscribe to the channel. Also, you can contact me via my email address, my Twitter, and also 2 addresses in Instagram. Thank you again, and bye!